Good evening, everyone, and thank you for attending tonight's event, Superbug Solutions. I'd like to start by acknowledging the Kaurna people, the original custodians of the Adelaide Plains, and the land on which the University of Adelaide's campuses here at North Terrace, Atway and Roseworthy are all built. My name is Hayley Parkinson, and I'm undertaking my PhD here at the University of Adelaide, and my research area is in multiple myeloma, which is a type of blood cancer. I'm going to be your MC for tonight. Antibiotic-resistant bacteria, often referred to as superbugs, have over the years been bolstered by overprescription and unchecked use in farming, and are now wreaking havoc on global health, claiming over 1.3 million lives annually. As we teeter on the edge of a post-antibiotic world, new solutions are needed to safeguard against infection. During tonight's presentation, you'll hear from how our researchers are developing groundbreaking medical therapies for superbug challenges like surgical site infections, non-healing wounds, and implant illnesses, and ensuring these breakthroughs transition from the laboratory into the real world. Our partnerships with veterinary scientists are also driving advancements in safer food production and animal wellbeing, and even everyday activities such as cooking are under scrutiny as we explore the complex interplay between everyday human behaviours and microbial threats. This evening, you'll hear from Dr. Katarina Richter, a research fellow at the University of Adelaide's Institute for Photonics and Advanced Sensing, and from Dr. Andrea McWhorter, a postdoctoral researcher within the School of Animal and Veterinary Sciences here at the university. So with that, let's get underway. Please join me in welcoming Katarina.
Thank you very much. Well, hello everybody. I'm really excited to be here. Before I give you a little introduction to my research, I was actually curious who is in the audience. Who is a scientist here? Okay, so everybody else is basically general public. That is amazing. It is really interesting to see because, scientists, we do research for you all. And it is really hard to reach the general public. So I really like the concept of Research Tuesdays because it brings science to society. And this is really close to my heart. So whatever we do it for the whole population. And this is close to my heart because I'm actually, I am a pharmacist by training. So I'm a healthcare professional. I studied pharmacy and pharmaceutical sciences in Germany, and have lived and worked in 7 countries by now. And I experienced a lot of infectious diseases, specifically with antibiotic-resistant bacteria, so-called superbugs. And superbugs really are around the world. It doesn't matter where you live, where you come from, you always are at risk to get infections with these superbugs. So my journey started in Germany. I did an honours degree in New Zealand where I developed nanomedicine actually for fertility control in possums. So basically developing the pill for possums. People in New Zealand really appreciated that because it's a major pest species there. And it really made me curious about science. This honours degree over there showed me that research is exciting. We are doing lots of things that nobody else has done before, and it can make a real impact in this world. So my curiosity got really sparked over there. But of course, it is like science is one thing, but like after a degree, what do you do with your life? So first I thought, I work a little bit, and I was curious to explore more of this world. So I actually went to Switzerland and worked in an emergency pharmacy. And over there, I had lots of patients with chronic infections. So basically, they had an infection that was never ending. They went to the doctor and got a prescription of just another antibiotic on a monthly basis. And with this, they came to us, to the pharmacy, and we gave them the best medicine we have, antibiotics. But clearly, that wasn't good enough for most of those patients. They still had infections, and it didn't get better. So it was very frustrating, not only for the patients, but also for us, because we really have limited resources. And that really gave me the idea to do a PhD one day, developing new treatments to help those patients with chronic infections. So this is basically where I found my purpose in life. I really want to help to develop new treatments, bring them to clinical trials and to products to save lives. Now, after Switzerland, I had a couple of other experiences in Denmark until I finally found my way to beautiful Adelaide to do my PhD over here. I came here 10 years ago and finished my PhD a couple of years ago, and I already have my own research group for 5 years by now, developing new treatments to fight superbugs. So my mission in general is to develop these new treatments and bring them into clinical applications. And this is a really hard thing to do because scientists, we do science in the lab, but sometimes it's really difficult to get this work out of the lab. So I'm really fortunate to have a great team behind me and lots of collaborators here in Adelaide and around the world to actually bridge the gap between science and society and bring new treatments into clinical trials or other applications to have an impact. So yes, our goal is to fight superbugs to the best of our abilities using different approaches, not based on antibiotics, so that the moment when we run out of antibiotics, we actually still have a treatment available for those patients suffering from superbug infections. Our second goal is to educate the society and tell them all about these kind of new innovations in the pipeline. Because as I said before, we do this work for you, so everybody should know about it. But yeah, superbugs. So they are one of the biggest threats to human health. They cause about 1.3 million deaths every year right now, and this is predicted to cause 10 million deaths per year by the year 2050. This will also be associated with an enormous amount of costs, so about $100 trillion. Superbugs will cost about $100 trillion to the global economy, and it will also push 28 million people into poverty. Now, one reason why bacteria become resistant to antibiotics is they live in biofilms. And biofilms, I like to see them as the slime castles of bacteria. So this is golden staph. You see those bacteria here. They stick to each other and they form a slime around them that gives them a protective layer, so like a castle. This is a wall of slime that protects the bacteria inside from anything that is happening around the world. So no matter if there's an antibiotic coming along, or whether there's UV light, or an immune system that wants to kill the bacteria, the bacteria within the slime are very well protected, and that means they're really difficult to treat. They can become easily resistant to antibiotics, and that also means they can require up to 1,000-fold higher concentrations of a treatment, like an antibiotic, to be killed than if we only have single bacteria. So it's really, really important to know this, but also to develop treatments that are not only effective against single bacteria, but specifically against biofilms. Now, biofilms, they're really everywhere. You find them from head to toe. They cause about 80% of infections in the body. For example, chronic lung infections like cystic fibrosis, implant infections, non-healing wound like diabetic foot ulcers, surgical site infections, or also hernia infections. These are just a couple of examples. Biofilms are really everywhere. So in most of those cases, golden staph is actually the bad boy. This type of bacterium really causes most of those infections. And just looking at one example here, hernia. So a hernia is when you have a hole in your abdominal wall where an organ or fatty tissue can squeeze through. And when this gets constricted, the blood supply can be cut off. And that means that the tissue outside the body can lack blood supply, and therefore the tissue can die. And this ultimately can cause sepsis. So people could die from it. this is actually quite common. So about 20 million people suffer from that. And to fix this and save the patient, surgery is performed. So hernia repair surgery is one of the most frequently performed surgeries worldwide. And in most cases, a mesh is inserted just to stabilize the abdominal wall, and then the wound can heal and everything is good. Well, in theory. This mesh, of course, is prone to bacterial attachment, and once this has happened, bacteria form a biofilm and can become resistant to antibiotics. So this is a vicious cycle and a real problem out there. And most of those bacteria are actually golden staph. So the treatment options are antibiotics, just one after the other, like I experienced back in Switzerland with all those patients. At some point, revision surgery is inevitable. But if you take out the hernia, if you take out the mesh, the hernia can recur. Or if you put a new mesh in, it is prone to biofilm formation, and we might get superbugs again. So it is kind of a vicious cycle. After surgery, the typical treatment is also, again, antibiotics, but this time for longer. So this all costs lots of healthcare costs. It contributes to a low quality of life of patients. And of course, it's a matter of time until multi-drug resistant bacteria, the superbugs, emerge, causing more problems further down the track. So therefore, new treatments are urgently needed. And I'm specifically looking at treatments that are not based on antibiotics, as said before, so that we have an alternative. Now, in the next couple of minutes, I'm very happy to share 3 different strategies with you we are currently investigating in my research group. So strategy number 1, well, for this, I let my other self talk. Let me show you what we've been working on in Adelaide. Superbugs like golden staph are resistant to antibiotics and cause over 1.3 million deaths every year. Thanks to funding from the Hospital Research Foundation Group, my team and I have been developing a new antibiotic-free super gel that fights back. Our patented wound healing gel can go wherever bacteria love to hide. It is liquid at room temperature, but when it reaches body temperature, it becomes solid and locks the treatment firmly in place to destroy bacteria exactly where they're hiding. It's non-toxic and promotes faster wound healing, meaning better outcomes after surgery. We are now ready to take our super gel to preclinical trials. So thank you very much for making this possible. Yeah, so this super gel is currently work in progress, but we're really excited to have progressed it so far. So what I'll show you in a second is basically a microscopy slide with golden staph. This golden staph is growing under steady nutrient flow, so that over time we actually get big blobs of biofilms, and they are at some point they are so big that they're actually blocking the channel of nutrients, so that everything is static and just growing more and more. So that is basically our control. You see the big blobs of biofilms forming over time, and then now the channel is blocked and it's growing further. Now, when we repeated exactly the same method, just supplementing the supergel into this growth medium for bacteria, it was kind of boring to watch because nothing happened, which is a great thing because we could show that we can prevent bacterial attachment and biofilm formation. So we have done lots of other investigations showing that this particular gel is not only effective against golden staph, but against also against a couple of other bacteria that play a major role in chronic wounds and surgical site infections. And we developed this kind of, they called it the super gel. We developed this super gel together with my PhD student, Loreen. Well, she just finished her PhD, so now she's a postdoc. Loreen, together with the University of Freiburg in Germany. And we actually made this gel liquid at room temperature and solid at body temperature. So it contains particular chemicals that make it liquid at room temperature. That means whenever you apply it, for example, on the surgical side or on a wound or even during surgery, wherever it is, you can inject it. It's liquid, it can fill all the niches, also where bacteria love to hide. And then when the gel heats up to body temperature, it is solid. So it locks all the treatments in place. That means we have longer time of our treatment to interact with the biofilms and the bacteria. That means higher efficacy. It doesn't go anywhere else, so we can prevent side effects in the body and therefore have better clinical outcomes. Now, the second strategy I would like to share with you today is in collaboration with a couple of researchers and clinicians in Adelaide. And this is based on a laser. So we are currently developing a medical device using a laser to basically treat infected wounds and surgical site infections. And my student Harriet has this as her project right now. She's specifically looking at this laser at a particular wavelength. So we are using UV light, but of a single wavelength. So as you know, UV light can cause cancer, but this is mainly for UVA and UVB. UVC is also part of UV light, but it's a short wave UV light. So it doesn't contain that much of energy. That means smaller particles like bacteria can be easily killed, while bigger particles like human cells, which are 10 times bigger than bacteria, they actually survive this without problem. And we are currently developing this laser device for medical applications, for different use in surgical site infections, for chronic wounds, for diabetic foot ulcers, everything where you could use a topical, like a local approach with this laser. And for the third strategy in our development pipeline, I would like you to think about lightning. Now, lightning is a natural phenomenon of plasma. Plasma is also the aurora borealis. Plasma is the fourth state of matter. So we have solid, liquid, gas, and then if we put more energy into matter, we get the fourth state, which is plasma. So that is a very rich energy state. It contains lots of molecules and fragmented ions, lots of different gases and radicals, and it's short-lived. So this high energy matter can be produced, well, in nature, but also you can do this for yourself when you microwave grapes or when you use a plasma generator. So this is how we do it in the lab. We're collaborating with a company called Plasma Treat, and they provided the plasma generators. So we thought about an easy application for humans would be a liquid. So we actually plasma activated water to make it antibacterial. And that means we spice up the water so much with lots of radicals and different other molecules that it still looks like water, but it has actually antibacterial properties. Now we made different plasma-activated water types. They all were made in different ways and they had different properties. And against specific like superbugs from the clinic, we tested them against those. And we found that one particular plasma-activated water type was really effective against those bacteria, so we only required a 25% concentration of this water. That means a dilution, like a 1 in 4 dilution, was good enough to kill superbugs. The other plasma-activated water types were not as good. Now, again, in this 3D biofilm model where we grew gourd and staph under the microscope, under steady nutrient flow, we saw lots of biofilms growing. And when we treated the biofilms with plasma water, we saw that the biofilms were shrinking and they were in other parts also detaching from the microscopy slide. We then added microscopy dyes to see how many bacteria are still alive and how many are dead. So here in green are the bacteria that are alive, and here in dead. In red, they're dead. So you could see that after the treatment with plasma water, we had the majority of bacteria being absolutely dead, and in the control, nothing was dead, everything was alive. So with this, with this new technology, we actually see lots of different applications. So we tested very good efficacy against different kind of superbugs and biofilms. We tested that it is not toxic in the lab and in preclinical studies, and we see a lot of different applications. For the human use, on the one side, for example, as a wound cleanser, or for dental applications, or even as an irrigation during surgery. But we are also branching out to other fields to use this plasma-activated water. So we actually got funding from the Medical Research Future Fund to go into pig studies in the near future, and ultimately into a clinical trial next year using the plasma water as a wound cleanser for diabetic foot ulcers. And that's going to happen here in Adelaide. We also see applications in the veterinary field to improve animal well-being, but also in the food area to improve food safety. And this is a project together with Andrea, who is coming up next, who investigated the effect of the plasma water against foodborne pathogens. So she's going to tell you all about it in a second. Of course, this is just a brief overview of the activities in my group, and that's not all based on me, obviously, so I have a great team behind me. Together we really achieve much more than as individual people, and together we fight for the purpose to bring new treatments to clinical applications and bridge this gap. So the translation of knowledge and innovations from bench to bedside is really our goal. So thank you to everybody who is helping me along the way. And with this, I would like to introduce the next speaker, Dr. Andrea McWhirter. She is a veterinary microbiologist who will give you more information about plasma water and other treatments.
Thanks. Thank you for coming to Research Tuesday this evening. I'm really excited to be sharing some of the research that my group has been doing, investigating how we can do a better job of controlling bacteria in food. So for most of us, eating food is not really a moment when we think too much about bacteria. We eat food because we're hungry. We eat food for enjoyment and for the opportunity to enjoy a diverse range of different culinary delights. Food is part of our celebrations and brings us together socially at different sorts of gatherings. Food helps us grow and keeps us healthy. But when we eat contaminated food, gastrointestinal disease can occur, which can have both short-term and long-term human health issues. Globally, foodborne disease occurs, we have about 600 million cases that occur annually worldwide. This affects 1 out of every 10 people over the world. In a recent New South Wales Food Authority study, 94% of Australians recognized food safety as being of significant importance over nutrition as well as potential health benefits of different food items. Yet we still have over 4.1 million cases of foodborne diseases that occur in Australia annually, affecting nearly 1 out of every 6 Australians each year. Foodborne disease can be caused by parasites, it can be caused by viruses and bacteria. And in the face of emerging antimicrobial resistance, food safety is and will continue to be a significant issue for continued public health. Of the 600 million cases of foodborne disease, nearly 350 million cases worldwide are caused by bacterial species. The 4 leading causes of bacterial foodborne disease in the world are Campylobacter, enterotoxigenic E. coli, Salmonella, Shigella, and then other bacterial species. The picture is a little bit different in Australia, with the vast majority of foodborne cases being caused by enteropathogenic E. coli, Campylobacter, Salmonella, and Clostridium perfringens. Over the past several decades, however, the outbreaks of Salmonella have been increasing in occurrence. And this is commonly associated with the consumption of raw or undercooked chicken, or raw or lightly cooked egg products. I'm part of the Poultry Health Research Group in the School of Animal and Veterinary Sciences at the Roseworthy campus. Our work primarily focuses on poultry health and understanding the persistence and control of bacteria that infect poultry birds and then move through our food supply chains. Australians love chicken and they love eggs. On average, we consume 49 kilograms of chicken meat and 262 eggs per person per year. And this is expected to continue to increase. Our national layer hen flock, so egg-producing birds, is over 22 million, and these birds produce 6.6 billion eggs for us each year. To meet the chicken meat demand, we have greater than 700 million broiler birds, or chicken meat birds, that were produced in the 2021-2022 financial year. For such a rapidly growing industry, there are a number of challenges, including the maintenance of a safe and secure food supply, as well as maintaining good poultry health and welfare. Two of the most commonly associated foodborne bacterial pathogens are Salmonella and Campylobacter. And the rest of my talk is going to primarily focus on these two organisms in both egg and chicken meat food supply systems. Poultry are natural reservoirs for both Salmonella and Campylobacter. Salmonella and Campylobacter can infect the gastrointestinal system of the birds and persistently amplify in their intestines. Birds, when they become infected, do not typically exhibit disease, and they subsequently can intermittently shed the bacteria into their farm environment, which can lead to contamination that transmits continually through our food, poultry and egg food supply chains. So why is it important to kind of understand the bacteria from a farm to fork perspective? So before I get started with that, I'm going to briefly describe the format of most of my graphs. Whoops. On the vertical axis, you'll see increasing numbers. And the larger the number, the more bacteria that will be present at a particular time point or in a particular treatment. And most of the horizontal axis will be represented by time or treatment. In a study that we did a few years ago, we investigated the prevalence of Salmonella on both layer hen chick rearing farms as well as egg producing farms to understand the dynamics of Salmonella on both of these farm systems. We found that in general, Salmonella was very low on our chick rearing farms, but in our production farms, we found fairly high levels of Salmonella in both dust and chicken feces. But the incidence of Salmonella infection on our eggs was quite low. This, however, is highly dependent on an individual farm as well as the farm system type. As you can see here, in this second farm, we also see Salmonella appearing in the dust samples. But one key thing to note about this farm is that the scale on the vertical axis is much smaller than the scale on the axis on the left-hand graph. And this peak of bacteria that you see here would translate to this blue line you see on the left graph. So sometimes we can have a high prevalence of the bacteria, but sometimes it can be low. So controlling bacteria on our farms has significant implications for both our transport of our food, but as well as storage and processing, and ultimately how the bacteria behaves in our food systems. To highlight this importance, we did some work investigating how salmonella can persist in a very popular raw egg-based food item, mayonnaise. We prepared in the lab, which most people hate it when I prepare mayonnaise in the lab, 2 different types of mayonnaise preparations. One was acidified with lemon juice and vinegar, and the other one was not acidified at all. We added salmonella to the mayonnaise preparations and then determined how long the bacteria could persist in the mayonnaise over time. From our work, we found that the acidification of the mayonnaise restricted the persistence of salmonella under refrigeration conditions, and no bacteria was detected after 12 hours after post-inoculation. Mayonnaise preparations that were not acidified, however, supported the persistence of salmonella over the entire course of our experiment. If you think back to our 2 different farms where we had both high levels and low levels of salmonella and the potential issues that might incur throughout our food supply chain, we also tested the differences in bacterial concentrations in mayonnaise preparations. In this experiment, we added 1 million cells of salmonella or 1 billion cells of salmonella to different preparations of acidified mayonnaise to understand how the number of bacteria can contribute to increased persistence in our food. And what we found was, not surprisingly, that the more bacteria that were in the food items, the longer we had viable bacteria remaining. In many of our food supply systems, we use food-grade sanitizers to control and mitigate both salmonella and campylobacter in the egg and chicken meat food supply chains. One of the most commonly used sanitizers is chlorine. And to test the efficacy or the ability of chlorine to kill bacteria, we first isolated a number of field strains or wild strains of salmonella and campylobacter from chicken meat samples that had not yet been exposed to any type of sanitizer. We then treated each of these separate isolates with chlorine and then monitored how the bacteria responded over time. Each of these lines present in these graphs represents a different salmonella or campylobacter isolate. And we saw that some bacteria were completely unaffected by chlorine, and the amount of bacteria that was in each of the samples persisted over the entire course of the experiment. But other isolates of bacteria were, in fact, killed fairly quickly following exposure to chlorine. So we need to develop new treatments beyond chlorine to minimize both salmonella and campylobacter in our food supply chains. These images are scanning electron micrographs of campylobacter that have been exposed or not exposed to chlorine. And normal campylobacter has a spiral-shaped morphology, whereas damaged or dead campylobacter appear round. When we looked at the chlorine-treated bacteria, we did find quite a number of still normal looking bacteria. And these bacteria remain viable and can recover following exposure to chlorine, which really represents significant risk for our downstream food supply chains. So from this point on, I'm going to present to you some of the work that we've been doing to develop new ways to control the bacteria on farms and in our storage and processing facilities. So first, I'm going to talk about a super gel. for use as an on-farm control strategy. So our super gel is also liquid at room temperature, which allows it to be combined with vaccines that are used to control both foodborne pathogens and poultry pathogens. Once the gel and vaccine combination is ready, we can administer it to chicks. After inoculation into the chicks, the gel solidifies, and then over time, bioerosion of the implant, the gel implant, releases the vaccine, and the bird's immune response can react to it, providing protection. To test the ability of the implant-based vaccine method, we tested the Salmonella typhimurium vaccine that's commercially available for use in poultry. So we administered the Salmonella typhimurium vaccine loaded in the implant, as well as using the standard vaccination method. One of the things I want to make really clear is the implant method allows the delivery of the vaccine using a single dose. But the standard vaccine delivery method delivers the vaccine over a 3-dose period. And the first point of call for this trial was to test the presence of antibodies in the birds to Salmonella. Antibodies are our first-line shield of defense against invading pathogens. And we found that at the end of the standard vaccine dosing procedure, that the antibody response of both the implant-treated birds and the standard vaccination birds was very similar. We also investigated the ability of immune cells to see what their combat readiness was. In this method, we isolate immune cells from the blood, and then we expose them to wild Salmonella. Cells that have been primed with vaccine are able to proliferate faster than cells that have not been primed with vaccine. What we found was birds that were administered the vaccine in a single dose in the implant ultimately had a higher proliferative capacity than cells from birds that received the standard vaccination. We're really excited because we're now able to move into research with an Indian vaccine company, which will allow us to further develop this technology and broaden its use with multiple vaccine strains. I'm now going to move towards the mitigation of bacteria in our chicken meat food supply chains. So as I mentioned before, chlorine is one of the most commonly used food-grade sanitizers. There are a number of other chemicals, including peroxyacetic acid and acidified sodium chloride, that have been approved for use for mitigating bacteria in our food supply chain. Now, I've stuck in some sneaky graphs here. The one on the left shows actually the reduction of bacteria on a piece of chicken meat, rather than the total amount of bacteria. When we washed chicken meat in different concentrations of acidified sodium chloride and peroxyacetic acid and compared that with chlorine, we found that both peroxyacetic acid and acidified chlorine were better at reducing the total amount of Campylobacter on chicken meat samples, with both concentrations of acidified sodium chloride providing the greatest reduction in the bacteria. We also evaluated their effects on Salmonella. So here we're back to the total amount of bacteria in each of the samples. And we find that, again, acidified sodium chloride and peroxyacetic acid were more effective than chlorine at reducing Salmonella on our chicken meat samples. We've provided this data to the poultry industries, and they are currently considering potential implant trials of some of these chemicals, but it's a significant change for them in their poultry processing because the use of alternative chemicals would mean significant re-engineering of some of their some of their systems. Along with Kati, we are also exploring the potential use of plasma water as a potential food sanitizer. As Kati mentioned, when plasma is added to a liquid, in our case water, it alters the physical properties of that liquid or water. So the plasma treatment induces the production of radical oxygen and nitrogen species and lowers the pH. And depending upon the gas that's used to generate the plasma, we get different ratios of reactive oxygen and nitrogen species, and the pH is lowered even more. To test the effect of plasma water on as a potential food-grade sanitizer, we compared the killing capacity of plasma water with normal tap water. And we were a bit surprised to see in these experiments that Campylobacter and Salmonella were completely killed immediately after exposure. And this is likely, again, due to the bacterial killing capacity of both the reactive nitrogen and oxygen species, as well as the low pH. We wanted to understand one of the mechanisms that plasma water kills the bacteria, and we can do that by measuring the light absorbance of the media that the bacteria is in at 280 nanometers. At 280 nanometers, we can measure the concentration of nucleic acids, such as DNA, in the solution. What we found was the plasma-treated water samples had 3 times higher light absorbance than the water treatment alone, suggesting that there was more DNA present in the liquid sample of the plasma-treated water treatment group compared with the control. So what does this mean? This means that the bacterial cell membranes and cell walls are lysed or broken open, releasing the DNA or nucleic acid into the solution. Bacteria that are in this state are well and truly dead. So we then moved to explore the possibility of using plasma-treated water as a potential chicken meat wash to control Campylobacter. To do this, we washed chicken skin paunches with both plasma water, chlorine, and water, and compared the amount of Campylobacter that remained after washing. And we found that plasma water and chlorine were both effective at reducing Campylobacter from our chicken skin punches. But unlike chlorine, where we found occasionally a few viable bacteria, plasma-treated water resulted in no culturable bacteria. So plasma-treated water has some really advantageous properties, one of which is that it's just water. And we are working towards developing a way to collect and filter and reuse the plasma water so that we can ultimately reduce the amount of water that's used in our commercial food industries. And finally, I just want to take a moment to talk about antimicrobial resistance in foodborne bacteria. So, especially in the poultry industry. So in response to the World Health Organization's Global Action Plan against antimicrobial resistance, the plan encourages the reduction of the unnecessary usage of antibiotics in both humans and animals. And thankfully, Australia in this regard is also a lucky country because our agricultural industries have responded positively to this global action plan. And in fact, in our poultry industries, antibiotics are only used in response to a positive diagnosis of bacterial disease by a veterinarian, and antibiotics are administered strictly under veterinary supervision. but we need to continue our antimicrobial stewardship and limit the antibiotic usage in all of our agricultural systems, so that we can help prevent further development of antimicrobial resistance. And with that, I would like to thank my teams, especially the poultry health team out at the Roseworthy campus. Without our team, we would not be able to do this amount of work, for sure. And I would also like to acknowledge Katharina Richter and her team for their support and collaboration. And I just want to say that at the end of the day, we're not just colleagues, we're not just researchers, we've become good friends and we really enjoy working together. And lastly, I just want to acknowledge some of our funding agencies. And last but not least, we are always very grateful to our poultry producers. Without being able to access their farms, doing a lot of this work would be completely impossible. But it is essential to helping mitigate bacteria in our food supply chains. Thank you very much.
Thanks so much, Katharina and Andrea. That was really fantastic. It's so interesting to hear about the things that you're developing and basically how you're going to save us from superbugs. So thanks so much. So we'll jump straight into question time now. So if you've got a question here in the audience, or if you have one online, you can type it in the box at the bottom of your screen. But for those in the room, if you've got a question, just raise your hand and we've got some roving mics that will come around.
Thank you. Katharina, I was very interested in your work. When you put the gel in, the super gel, does its killing power persist, or does it deteriorate over time and need to be replaced? Or once you've put the gel in, is it done for good, there for good?
It will deteriorate over time, but because the gel also has some wound healing components, we're actually having a double whammy here. So on the one side, it has antimicrobial properties to fight off superbugs. On the other side, we're improving wound healing so that the surgical wound or the diabetic foot ulcer or whatever wound we are treating actually can heal better and faster.
It's only taken around 80 years for our superbugs to be many superbugs against the antibiotics. Is there any research being undertaken as to how long we've got until the bugs start to treat your gel as champagne?
I like champagne, but I don't like the thought of, superbugs taking over. So this is a very valid point, and I think every researcher around the world who develops new treatments against superbugs, we all do studies to investigate whether bacteria on the one side become tolerant or even resistant to particular treatments. So these studies are important and they are being carried out. Whether the studies, are real, we could right now see that there is, so far at least, there's no risk for resistance, but you never know, right? So this is all lab-based, and sometimes whatever we figure out in the lab doesn't really mean this is exactly how it's happening in the real world. So after all, when whatever kind of new treatment you use, you need to monitor for resistance. And you need to identify very quickly whether bacteria adapt and become resistant, and then also pass on these resistances to other bacteria or not. After all, we also, in terms of treatment regimens, we are better off to actually use different approaches all together. So combining different approaches to have the maximum effect so that we can prevent resistances.
Thank you. It wasn't quite clear where you saw the plasma water or acidified chloride used in the preparation of chicken meat. Do you envisage a whole chicken carcass being washed with one of these things? And secondly, plasma water
full of free radicals sounds pretty unstable to me. So I presume you can't make it and stick it in a bottle and use it next week. So what about the stability when it comes to practical applications?
Yep. So the idea of using plasma-treated water as a chicken meat sanitizer would be for implementation at some point in our chicken meat processing plants. So at our processing plants, we currently have large spin chillers that the carcasses travel through, and those contain chlorine to reduce the not only food spoilage bacteria, but also Campylobacter and Salmonella and other foodborne pathogens. In other countries, they utilize sanitizers at the end of processing as well in sort of a spray type of environment. And so we're working on developing the most effective delivery method for either to contribute to the reduction of bacteria following chlorine treatment or on its own. In regards to your comment about the plasma-treated water, yes, the reactive oxygen and nitrogen species are very labile, very short-lived. When they come into contact with an organic surface, they are inactivated very quickly. But we have been able to store the pure water in sterile plastic containers for upwards of 6 months. And there's no change in the presence of the reactive oxygen and nitrogen species or any change in the pH, which suggests that there are other alternative applications that could be used, for example, in commercial or home kitchens. But we're going to have to continue to collaborate with Plasma Treat in order to evaluate some of those treatment options.
Hello, thank you for your talks. So to what extent do you think there needs to be an emphasis on reducing our dependence on meat consumption to solve this problem ultimately? Do you think that the approaches that you're taking will ultimately be the only way that we can fight this problem, or do you think we'll have to actually reduce our meat consumption?
That's a very good question. And a little bit of a difficult one, mostly because the vast majority of our antimicrobial resistance is emerging from our health and medical fields. And there are becoming high levels of antimicrobial resistant bacteria that are emerging in our sewage treatment systems. And if you recall the kind of web I showed at the end of my talk, each system is not disconnected from every other system. And so we can have movement of antimicrobial resistant bacteria throughout all of our health, medical, environmental, and food production systems. And so if you were to water vegetables that contained bacteria that had antimicrobial resistance, well, those bacteria can also persist on leaves of lettuce or other vegetables. So in terms of just focusing on reducing our meat consumption, that's a difficult question to answer, mostly because all of our systems are interrelated and all of our systems are affected by each other.
Thank you for very interesting talks. Chlorine is obviously very toxic for people if they're inhaling it while the treatment's occurring, so there's a lot of protection and safety going on. Is plasma water, do you know the effect of plasma water on epithelial cells? Is it a lot safer to use in that sort of environment and those sorts of things?
Yes, so in our food processing plants, we use chlorine at fairly low concentrations, so 8 parts per million, which is generally lower than what is used in public swimming pools or your swimming pool at home. Yes, the production of aerosol mists and gases do represent a significant risk to workers, but there are a lot of safety mechanisms in place to protect people. In terms of plasma water, those types of experiments have not yet been conducted, especially on an industrial scale. Kati has done some work in the lab that demonstrates that the plasma water does not have any negative effect to cultured cells.
So we tested different types of human cells in the lab, and we saw no negative effect of the plasma water on the viability of cells. We also did animal studies. Please don't punish me for that. So we did infection studies in different animals, including mice, and a pig study is coming up. Because this mimics human skin better than a little mouse. But we didn't see any kind of toxicity, so neither on the skin cells of the animals, nor on blood cells, nor on any kind of organs. Like, there is no side effect so far.
But you'd have to do much further research in order to confirm that.
I mean, our clinical trial, which we're going to do next year for the treatment of diabetic foot ulcers, this is not a trial for efficacy of the treatment, it's a trial for safety. So that will be a first-in-human study, and once we have this data, we can certainly be sure of any kind of potential toxicity.
I think we have a question from our online audience, so that's just coming from up the back.
Yeah, this one's for Andrea. In view of the variation among strains you noted for one experiment, do you now test the efficacy of experimental treatments, for example, plasma-treated water, against the mixture of strains or the mixtures of species?
Yes, to both questions. So for this presentation, I simplified a lot of the data that we've obtained, but because we saw a high degree of variation in the bacterial response to chlorine, we did test a number of Salmonella and Campylobacter strains for the bacterial killing capacity of the plasma water. And unlike with chlorine, we found that the plasma water killed them all dead regardless of the strain that we used.
I actually have two questions. One is, how important do you think it is to reduce overprescription of antibiotics for the overall cause? And secondly, is there a worst case scenario strategy where you treat it like an infectious disease or something, so you try and deal with it like through isolation or something like that?
I didn't fully understand the second question, but in terms of reducing the use of antibiotics, that is essential. I mean, data from actually around the world has shown that the moment we reduce the overuse of antibiotics, also the number of resistances go down. So antibiotics are still our most powerful weapons against bacteria. So we need them, and they're very effective, but we need to use them wisely. So whenever you have a particular infection, of course, it would be way smarter to first do a culture, identify the enemy, and then take an antibiotic that is specific against this bacterium, and not a broad spectrum antibacterial that basically kills all the bacteria, including the good bacteria in your gut. So a targeted therapy would be way better, but I also understand that, in the clinic, sometimes, you just don't have the time to take a swab, to culture the bacteria and figure out resistance profiles. So if the patient, if it's about life and death, then you need to give a treatment right away. And for these, emergency situations, of course, you have to use a broad spectrum antibiotic. There is no other way at this point, at least. But in general, it is, you need to monitor how frequently you prescribe antibiotics and if they're really necessary. So the severity of disease is really important to understand, and based on that, the doctors have to decide whether to give an antibiotic or not. Did this also answer your second question? I don't remember.
I guess in the scenario where suddenly we can't deal with them, I realise that these things are sort of all around the place, and isolating them is very difficult. But in terms of like if a person gets golden staph, I mean, I guess maybe I'm just indicating my lack of knowledge here, but I presume there's like protocols of sort of isolation, or you know, can you sort of really try and hedge it in and have some kind of really hard, strict regime that, you know, is there a fallback? You know, obviously not something you want to do. Maybe it's something you scare people with, you know.
Yeah, so basically the idea is to stop the spread, right? So once you have a superbug, whatever superbug it is, you certainly don't want to pass it on to anybody in your family, your neighbors, your colleagues, whoever. Yeah, hygiene is really important. And yes, isolation. I mean, COVID taught the whole world that simply washing your hands can make a huge difference. And also, if you're sick, just stay away from others. Maybe, you know, if you like, you know, when my husband had COVID, we slept on separate sides of the house and we used 2 different bathrooms, and, you know, just to protect the other. And it actually worked. So, and that was with a virus which was way more contagious than most bacteria. So it is about, you know, understanding about these kind of preventive measures, but then also doing them. So not only knowing about them, but also implementing those preventive measures in your life. Whenever I'm a big fan of public transport. Whenever I come home, I always wash my hands. That's the first thing I do. I mean, I don't know whether this truly helps, and you know, you never know whether you have any kind of superbugs on your hands or on your shoes or whatever. But you know, if you are aware of bacteria being really everywhere around you, then it's in your own hands to actually do these kind of little things to protect you and everybody around you.
All right, in the interest of time, we might wrap things up there. So if everyone can please join me in giving a big thank you to our fantastic speakers. And of course, thanks to all of you who have come here tonight, and those of you who are watching at home. Thanks again for tuning into another Research Tuesdays. Be sure to sign up for our mailing list to get information on all our future events. So thanks for coming, and see you next time.